get going. Welcome to the Designing Therapies for HD Lessons Learned from Other Disorders workshop. My name is Stephen Martin. I'm the Research Center Director for East Central Ontario, which is Peterborough, Lindsay, Barrie, Oshawa, Ajax. That's my region. Okay. Before we get started, a couple housekeeping items. The session runs for 45 minutes. There'll be lots of time for questions before it's time to go. If you need to step out, or if there's something that is bothering you about today's session, just right on the outside of that door. That room is called the chill room, where you can have some quiet time if you need it. Okay. So let's get going. I am thrilled to be joined today by our speakers, recipients of HSC Navigator Award, Dr. Shaun Sanders, Dr. Melanie Alpaugh, and Dr. Dale Martin. Dr. Sanders and Alpaugh are both assistant professors at the University of Guelph, and Dr. Martin is associate professor at the University of Waterloo. Is there competition on that?
I can tell you over.
Yeah. We want to thank the three of you for joining us to have this conversation today. Thank you.
Great, thanks so much, Stephen. Thank you all for coming today, and thank you to the Huntingtin Society for inviting us to come and tell you a little bit about the work we're doing. So I'll talk a little bit about what we're doing with the Navigator grant that was received last year, collaboration between the three of us, and I'll also tell you a little bit more about other things happening in our labs. So. To start, we thought we would tell you a little bit about ourselves. So I did my undergrad at UBC in Vancouver, and while I was there, I started doing research in a lab, and I was working on a neurodegenerative disorder called Tay-Sachs, which affects young children. And so I kind of got into research that way, and then went on to do my PhD with Michael Hayden at the CMT in Vancouver. And that's where I got introduced to research on Huntingtin disease. And while I was there doing my PhD, Dale and I met. So we are actually married and collaborators in life and in work. So we met while we were both at UBC in Michael Hayden's lab. And from there, I went on to Philadelphia to the Temple University of Lewis Katz School of Medicine, and I actually kind of moved away from PhD work a bit and was working on neuronal injury. And this kind of connects to what we are doing now with the Navigator grant. So, and then in 2020, I started my lab at the University of Guelph in early mid 2020, and so it was definitely an interesting time to start a lab. So it's been 5 years now, and so I am the bridge between the three of us here, so being married to Dale, and Melanie and I share lab space as well. So yeah, so my lab, we're interested in general in how things get where they need to get in brain cells, in neurons. These are really big, complicated cells, and so it's not a simple feat for all the machinery within the cell to get where it needs to be. And so that was kind of the aspect that I was working on in Huntingtin disease in Michael's lab, and then in neuronal injury in Philadelphia. And so we're kind of still working on that aspect. So this is my group, and here we were at an axe throwing event, and the Navigator grant is funding Julia here. She's our lab tech, and so she's doing a lot of the work in my lab for the Navigator grant. And then we also have an undergrad, Micah, who is helping Julia with all the data that's being collected, trying to analyze it all. So they're both hard at work on the work that's being funded with the Navigator grant. So now I will pass it off to Melanie to introduce herself.
Yes, so my research journey started at the University of Alberta in Edmonton, where I did both my undergraduate and graduate training. So in undergraduate, I went through a number of different research fields. I started by doing mutation screens in fruit flies. And then after that, I started doing psychology studies where participants came in and answered questions about personality traits. Then we did some EEG, recording activity from the brains, but none of that really matched where I wanted to be. And so then just before I started my graduate studies, I actually began working on Huntington's disease. And I have been working in the field ever since. So that was back in 2010. There we go. Like, that sounds wrong. And then after I completed my degrees in Edmonton, I continued my tour of the coldest cities in Canada. I decided to move to Quebec City. Yes, I did eventually learn and move to Southern Ontario, but I spent about 5 years in Quebec City learning French, surviving COVID, speaking a second language in a mask, but continuing to research. Do not recommend for anyone who's considering trying it. And I also continued to do Huntington's disease research while I was there. And then in 2022, I was fortunate enough to get a position in Guelph, where I actually joined Shaun's lab space, as she mentioned. And so now I like to say we have the second highest concentration of Huntington's disease researchers for labs outside of Vancouver, because there's 2 of us at Guelph. So my research has broadened, and we really look at a number of different diseases. And I've highlighted some of my students here. So this is one of our semesterly lab activities. So we were doing paint and sip, but we of course did it on campus because it was most convenient. So we absolutely did not have real alcohol on campus. But I've highlighted a few students here that that have work related to the data that I'm presenting. Specifically, I'm going to tell you a bit about Christiana, because she was funded by the Huntington Society of Canada and is doing some work on the DLK project. So I'll pass off to Dale.
Thank you. So yeah, I have a similar overlap with both Shaun and Melanie, obviously with Shaun. So I started off on the East Coast in Dalhousie. I did an undergrad there. I did cooperative education, so I did work terms, and so I worked on yeast and multi-drug resistance and stuff like this. But one of those work terms actually took me to the Bay Area, to Oakland, where I was working in cardiovascular disease. And so that actually will be some overlap with Melanie, and also relate to some of the projects that I'm going to tell you about a little bit later. And then so I went over to Alberta, and this is where I did my graduate degree. Overlap with Melanie, maybe. We think we met, but we don't know if we met.
We think I dropped off some samples
Yeah.
to the lab.
Yeah, I think she just started when I was just finishing, but I just started. I found kind of serendipitously this modification of Huntingtin that was really rare, and then so that's where it got me kind of really interested in Huntington disease. So then I decided to go do a postdoc, and that's how I met Shaun in Michael Hayden's lab at CMMT. So there I was studying Huntington disease. But then Shaun and I got married, and Shaun went over to Philadelphia, and I didn't want to be away from her, so I also started working on DLK with Shaun, and so that's what our Navigator is about, is this actual project. That kind of stemmed from there as well. And so I started about 2018, a little bit before the pandemic, so we also got shut down, but we have a really fantastic group of people now. This is us. We have a brand new fancy lab, so they use us to like all over campus to be really famous here. But so there's quite a few different people. I'll highlight them a little bit later, but there's quite a few different HD projects that we have going on in the lab. And then I'll also highlight, so we have this artist that is Kaylin, and one weekend she came in and drew Rubio, so we can never erase our whiteboard again. And then Ruby came in to visit, so she's kind of like our unofficial mascot, so that's our Shaun and our dog. And so in my lab in particular, we study multiple neurodegenerative diseases. We focus primarily on Huntington's disease as well as ALS, so amyotrophic lateral sclerosis. And the reason we do that is what we're trying to do is we're trying to find where there might be overlap in various neurodegenerative diseases. So like, what is the pitch point? What are the shared commonalities between these diseases? And then so if we find a therapy for one, can we potentially apply it to the other? Or at least can we learn something for one and apply it to the other? And so in that, with that idea in mind, one of the things we're doing is we're trying to repurpose like FDA-approved drugs that we found for another disease. We're applying that to Huntington disease. One of the things that kind of why I'm on this navigator as well is because we also have the mouse models of Huntington disease for this. And then another thing that I'll talk about towards the end is one of the things that's kind of common among these neurodegenerative diseases is the buildup of mutant proteins. And so we're trying to figure out how to remove them and how to destroy them essentially. And then finally, we're also working on novel delivery methods. So trying to get drugs into the brain is very challenging. And so some of the stuff that we're working on is how to get that across the blood-brain barrier, which is really Melanie's topic. So I'm going to hand it over to Melanie.
Yes, so one of the main things that my lab has been working on is understanding how the blood-brain barrier, which is a really important communication point between the body and the brain, is involved in a variety of different disorders, including neurological disorders and cardiovascular disease. And part of this, and one of the things we'll talk about today, is I've developed a model system that really allows us to understand what's happening there, and it also lets us test some of these different delivery methods that Dale is developing, because we can put them into essentially an artificial blood vessel and see if we can get them into the brain. And then overall, my lab is also more broadly understanding how illness like heart disease may be influencing our brain, and how maybe we can target heart disease to help slow what's happening and help delay the problems that are ongoing within the brain. And so then I will pass off to Shaun to tell her more about the project we worked on all together.
Yeah, so the Navigator grant is, as I kind of mentioned, an idea that started when I was in my postdoc and I was working on like spinal cord and brain injury. And what happens is there are these degeneration signals that are sent. And this is actually a common pathway, like what Dale alluded to, that happens in many different types of neurodegeneration. So from Alzheimer's to Parkinson's, ALS, as well as in brain and spinal cord injury. And so the brain cells, neurons, are these really large, complex cells, and they receive information from other cells on dendrites. These are the receivers. And all of that information is processed within the control center, the cell body. And then the cell will decide to send the signal down to the next cell. And those outgoing signals are sent down these long cables and then eventually transmitted to the next brain cell. And within these long cables, there are highways, and different cellular components get sent down these highways. And these are really, really big cells. So some of these axons in the body, there's axons that project from the brain all the way down the spinal cord. So these can be really long, up to 1 meter long in humans. So these are really, really big, really complicated cells. And if there's a problem happening out here, the brain cell, needs to have a system to alert the cell body so that we can get a response. And so when I was in Philadelphia working out on my postdoc, I was working on this protein called DLK, or dual leucine zipper kinase. And it's a stress sensor. So what it does is it gets sent all the way down these long highways to the very ends of the axons. And it just kind of hangs out, and it's just there waiting as an early detection system, waiting for a signal that something is going wrong. And what happens when something goes wrong, so the most classical example when we're studying DLK is brain or spinal cord injury, nerve injury. DLK gets activated. And when it gets activated, it sounds the alarm. So this alarm then will get sent, turned on, in response to, as I've already alluded to, nerve injury, and that happens both in the periphery, so like in our limbs, as well as in the central nervous system within the brain itself. But DLK has also been shown to get activated in neuropathies. So these are nerve damage that happens during, say, diabetes or with chemotherapy treatment. We can get neuropathies that develop, and we know that DLK sounds the alarm in that case as well. And then more recently, it's been shown that DLK is also getting activated in other neurodegenerative diseases like ALS, Alzheimer's disease, and Parkinson's disease. But we don't know anything about what's happening in Huntingtin disease if DLK is getting activated and sending that alarm signal. And so what happens once DLK is activated, it sounds the alarm, and this alarm signal goes all the way back down these long highways back to the control center, that main cell body, where it causes the downstream signal, these prodegenerative signals. So what happens is we get a change in the levels of the proteins that are being made within the control center, and that can eventually elicit degeneration, so cause degeneration or death of the neurons. And clearly in Huntington's disease, we wanted to prevent that from happening. And so work that's been done in these other neurodegenerative diseases shows that if you block that alarm, you block DLK activation, you can prevent degeneration. So is the same thing happening in Huntington's disease? And so that's the goal of the Navigator grant. And our data so far suggests that this alarm is being sounded in HD neurons. And so what we're going to start doing now is block that DLK activation, block the alarm from being sounded, and determine if that can prevent some of the degeneration that we're seeing in Huntington's disease. So prevent or slow the death of brain cells. And so that's the goal of the Navigator grant, and we're still working really hard. As I said, Julia's been producing lots of data, and we're analyzing it all, and in collaboration with Melanie and Dale, continuing to answer this question over the next year of the Navigator grant. So now I'll pass it off to Melanie to tell you a little bit about the work in her lab.
Yes, so with the DLK project, one of the main contributions of my research group is that we have cell culture models that are very valuable for these studies. But we also were able, with support from the Huntingtin Society of Canada, to add an extension onto this project. So this summer, I had a very talented undergraduate student, Christiana, who actually is now in medical school. She got in after her third year, and she's hoping to become a neurologist in the future. So that support, yes. So we're trying to get her into the Huntington's field from the very beginning. And so what she was helping us with this summer is we actually were able to get access to some human brain samples. So she was processing those and setting them up so that we can do some more protein analysis to actually see if these changes that we're measuring in the cell culture models and mouse models can actually be observed within the brains as well. So that's what we're doing, and this was the extension that we were able to add on to this project. But more globally, my research interests are really about the interaction between the brain and the heart. So if you look into neurological diseases, what you can find is oftentimes if you have changes to your cardiovascular system, if you have heart disease, if you have a heart attack or hypertension in midlife, that can actually make it more likely for you to have a neurological disease later on. So Alzheimer's disease in particular is very strongly associated with changes to your blood system, to your circulatory system. And also, if you have certain neurological conditions like anxiety or depression, you're also much more likely to develop heart disease. So these two systems really influence one another. And so if you actually look at the blood supply to the brain, this starts to make quite a bit of sense because we have a lot of cells in the brain, like a lot of cells. And all of these cells have many different activities they need to do, and that requires constant energy input. And so the blood has to supply that energy to those cells. So you need to have all of the oxygen, you need sugar, so that your brain cells can do what they're supposed to do. So if you have a disruption to the blood flow, if these blood vessels aren't working properly, then this can make it much harder for the cells in the brain to fulfill their roles. Additionally, because the cells of the brain are quite sensitive, the blood vessels are very specialized in the brain, and they're specialized so that they can protect the brain. So they need to make sure that the brain gets all the nutrients it needs, but they also need to make sure that things that are in the blood that are bad for the brain cells don't get in. So there's a lot of proteins that exist in the blood that are very important, but they can be toxic to brain cells. So there are a number of different cells that work together to help make sure that there is a barrier, but that the brain still gets all of the things it needs. So this becomes a very important point of communication between the brain and the body. and this is called the blood-brain barrier. And so my lab is very focused on understanding how the blood-brain barrier contributes to brain diseases and how having heart disease or other cardiovascular diseases can make it more difficult to function. So we do this using a number of different systems. I'm not going to go into this in too much detail. We do some animal work where we're looking at heart attack, what heart attack does to the healthy brain. We're working in a mouse model of Huntington's disease. We're looking to see what heart attack does to a brain when you have a genetic disposition to Huntington's disease. We're also looking to see if we can get rid of hypertension in these mice to try and make delay when we can start seeing changes associated with Huntington's disease. But what I'm going to talk about most today is this model system we've been working in, because this is most relevant to the DLK project and is also one of the key collaboration pieces between myself and Dale. So what I did during my postdoc is I decided that we needed, well, my supervisor told me that we needed to make yeah, I stand by it. So we wanted to be able to make this system where we could really understand in culture what is happening and how all these cells are working together. So we used stem cells. So these, you can take skin cells from any adult and you can turn them into stem cells, which means we can get them from those who have different backgrounds. So we have Huntington's disease gene carriers where we have these cells from, also individuals that live with other neurological conditions, and also those that don't have any changes within their brain, so that have a brain that's functioning normally. And so then we can differentiate them into the cells of the brain, so we can produce neurons, we can produce astrocytes and all those components of the blood-brain barrier, and then we can put them all together into a very special plate. So we break them up, so we have our endothelial cells, then we have a structural component, so that it actually will keep everything separated. We use a gel for this. And then we have our cells of the brain, so the central nervous system, so our astrocytes, neurons, our pericytes. And when we put them together, it looks something like this. And this actually allows us to make a blood vessel in a plate. So we can actually make a three-dimensional vessel in culture. And so then we can do a number of different things with this. We can see that it really does form a three-dimensional structure. We can see that our neurons are able to survive. And we also can test and see if it has the functions of this barrier. So if it's able to prevent different factors from getting from these endothelial cells into the brain. And we do this using dyes, so we can add the dyes on the top, and then we can leave it for 40 minutes and see where it ends up. And when we do that, we see it stays exactly where it's supposed to. So we have functional properties of the blood-brain barrier. And so then we can use this system to answer many different research questions. We can do this in many different diseases, and we can understand shared features across different neurological and neurodegenerative disorders. And so we can also test some of these compounds that Dale is studying, and we can look if we can get them from the endothelial cells down into the brain. And with that, I'm going to pass off.
Awesome, thank you. Yeah, so as I mentioned kind of earlier, we're working on ALS as well as HD. Partly, like I said, some of the common features, regardless of neurodegeneration, is that basically the proteins, like so mutant Huntingtin, they kind of clump together, and so we see those as aggregates. So those aggregates may or may not be toxic, kind of depends on the disease, but they're indicated that something has gone wrong with getting rid of these destructive proteins. And so one of the things we want to do is also target them. And so you can kind of see, actually on this screen, I don't think you can see it, but you can see kind of like how there's little tiny aggregates, how the blue, this is Huntingtin in a neuron, and you kind of see how it kind of clumps up and it goes to the wrong place. And so a lot of what happens before they aggregate is proteins go to the wrong place and they start doing the wrong thing in the wrong spot. So we're trying to fix that as well. And then also, ultimately what this is we're trying to lower the levels of these proteins. So kind of what we see in HD is the main way that people are targeting mutant Huntingtin is trying to lower its levels through multiple different ways. So far we've seen inside, we've seen what the neurons look like. This is basically kind of like what would be happening inside the cell. This is your nucleus. This is what has all of your DNA. The DNA here is kind of like what would encode for Huntingtin. This would be RNA, so DNA codes for RNA. It's basically the messenger, or think of it as like the engineer that has to read the blueprint. And then that was going to make the proteins. So the proteins inside your cells, they're what are driving everything. They're the enzymes, they're the things that are kind of making things happen. They break things down, and they also make stuff. And so there's a lot of different ways that we can target mutant Huntingtin. So I'm not going to talk about all of them, but you probably heard a lot of different ways to target the DNA itself. And a way a lot of people are approaching this is targeting the actual RNA itself. So you've probably heard about antisense oligonucleotides or tominersen, these kinds of things. These are these small molecules that basically, they're like DNA. They bind that RNA, and they cause that RNA to be destroyed. So we have 2 copies of everything, of all the genes. So like in Huntington disease, you would have one copy that has the mutant and one mutation, and then one that does not. So in this case, that small molecule can bind that messenger for the mutant and cause it to be destroyed. So it's no longer there. And so that's what we're trying to do. And then the idea would be that be specific and not hit that wild type Huntingtin or normal Huntingtin, and then that protein is actually going to be made. The problem here, and again, it's just a caveat at this point, but these don't cross the blood-brain barrier. And so this is why those ASOs have to be injected directly into the spinal cord, is because that way it can go directly into the brain. And so we want to try to come up with something that's a little less invasive, and so basically patients could just have the injections whenever they need it, that kind of thing. And so we thought that we needed a Trojan horse, something to sneak across the blood-brain barrier, right? And so this ties into a lot of what Melanie does, because by understanding the blood-brain barrier, we can also understand how things cross it as well. Right, so we were just trying to find out, proteins that we already know are in the brain, and maybe come from another organ. And so this is why we came up with APOE1, and this is why it's important what I was telling you with the work I did in Oakland, where I was working on cardiovascular disease. I was looking at cholesterol transporters, and APOE1 was the protein that I studied. And basically all you need to know is this is what makes up your good cholesterol, HDL. So if you go get a blood work and they tell you about your cholesterol, they're telling you about your HDL. This protein is made in the liver, and then it has to go everywhere else through the body, and what it does is it brings fat back to the liver. So it's collecting fats everywhere and bringing it back, and that includes the brain. So we know that it has to actually cross the blood-brain barrier to get into the brain. And so this became our Trojan horse. So we basically just took what was already there, and then we got it to bind the antisense oligonucleotides. And so this will be a little bit hard to see, but this is what they look like with an electron microscope. So this is like it's not super resolution, but it's really looking in. They're really, really small. They just kind of look like jelly beans, but you can almost think of them as kind of like a cheeseburger, right? Like the protein is the meat patty, and then these are the buns, basically, which is fats. And so we did find that, at least in mice, that these could help deliver ASOs by IV, so like intravenous, but also intranasally. So there's a couple different ways that it was working much better. And so I want to also highlight, I should have put his picture here, but Nick Perron, if you know him, he was a postdoc in Michael's lab. He was the one that also thought this was a good idea and helped me run with it, and he did a lot of this work himself. And so I'm not going to go through this because Melanie kind of already did it, but basically we are able to show that it does go into the brains of these mice and lowers mutant Huntingtin. The thing that we're working on now in my lab, and I'll highlight some of the students that are doing that, is what we try to do, and we did this with Nick as well, I should say, but what we did is we trapped HDL in this form, so it no longer wants to collect fats for the rest of the body and stuff like this, and more of it goes into the brain. And so that's also what we did to modify these. And so now we're trying to superload them essentially with ASOs, so we can try to really knock down way more mutant Huntingtin. So if we kind of come back to the next step, like I said, another target that we can actually do is the protein itself and try to get rid of it. And one of those pathways that we're trying to modify basically or exploit is something called autophagy. Autophagy means self-eating. And so your cells actually break down some of these mutant proteins or things that it doesn't need and it recycles them. And so that's basically, so if you look at these kind of right here, imagine that this is a full actual sphere. Basically, they engulf these mutant proteins and then takes them to the trash can of the cell, which would be kind of this like purplish, slightly chocolate, so that's purple. But basically, it's going to help to create them. I'm also showing this not just because we have the cover of this, but that's actually, I stuck Shaun's brain into the back of this. So that's Shaun's brain, the cover of the journal. Anyway, so basically what you need to think of, the simplest way to think of this is we're trying to take those mutant protein, not just the ADN, but mutant Huntingtin, and we're trying to send it to the trash can of the cell. And so a lot of the small molecules that we're working on, we're working on trying to direct Huntingtin to those trash cans, and then basically test them in Melanie's systems to see if they are actually going to cross the blood-brain barrier before we go into a mouse model. And so these are actual neurons from these mice. And so a student in the lab basically stained them. And so you can see where it's a bit of post-processing where it's nice and smooth, but these are the neurons that Shaun was kind of showing in the cartoons. And you'll see the colors kind of disappear. And then when you see just red, that's just mutant Huntingtin in this mouse model. It's actually all Huntingtin, I think, here. And then so this is the actual nucleus. So again, that's the DNA that's in the center there. And then as you'll kind of see, there'll be a magenta color come up. That's the trash cans of the neuron lit up. And then the green is a protein that helps take all those proteins to the trash can. And so we'll just zoom in one more time. And what it'll show you is, again, kind of like a nice close-up of one of these kind of areas where all three of those proteins or all three of those are there. So this is showing the protein that takes things to the trash can, and that's showing mutant Huntingtin, so kind of in the trash can there. And so that's what our small molecules are trying to do, and we're trying to do that there. And so this work is really done by three really talented students that were in the lab. Yasmin's actually coming back. She just finished her master's. If you want to see more of the stories here, the QR codes are to use her correction. She was working on the nanoparticles to optimize them for the delivery, and so she was funded for our HSC Summer Fellowship. And Anthony is one of the students. He came up, like he developed his own drugs essentially that we want to try to test. And he did this all through computational analysis. And so this we're trying to get into the lab now. And again, we're going to try those in all these systems. So with that, again, so those students, the work that we're doing, of course, could not be funded, or we couldn't do this work without the Huntington Society of Canada, but also I'm sure many of you have donated to the Huntington Society of Canada, so thank you as well for contributing to the students, the work that we're doing. And also I just want to highlight all of the patients as well, because we can't do this work without patients. And then also I just want to highlight a little bit, there's a lot of different funding agencies here, and like we said, all these different diseases, and so it takes many different approaches to find these kinds of discoveries that we're doing. And thank you. But
Yeah.
anything else?
No, that's great. Yeah,
Okay,
thank you very much.
awesome. Thank you very much.